All right, we are now at 1:15. I'm sure we'll have a couple more people trickling in as we go. First, I want to thank our sponsor for the conference as well as the session, GRIN Therapeutics. And in this session, we're actually joined by Dr. Michael Panzar, who's the Chief Medical Officer for Nervati Neuroscience and part of GRIN Therapeutics. So we're very excited to have him here to join us and present on this session. So this session is titled Insights from GRI Clinical Trials. We're joined by Dr. Panzar, who's going to speak first. And then we also have Dr. Angeles Garcia Cazorla from the Children's Hospital in Barcelona, who's going to talk about their clinical trial. Thank you so much.
Great. Thank you very much, Megan. And thanks to GRICON for giving me the opportunity to come here to present today. I'm going to talk about Verdiperdil, but I'm going to talk a little bit more broadly about clinical trials. Just a bit about me. I'm a neurologist and have been involved in developing neurology drugs now for a little over 20 years. Never in this therapeutic area, so it's exciting. And to be here, last year's meeting was my intro to this community, and it's been just a fantastic year as we've moved forward with our learnings about Verdiperdil, as well as getting to know the community over the past year. So I thank the community for that, and I'm looking forward to hopefully many more useful, a great partnership going forward. So this is the outline of my talk today. I'm going to talk about GRIN very briefly, but also we have a lot of questions about clinical trials all the time. And, there's every time I've been involved in a clinical trial in a new area, there's an educational process. There's a learning that goes on about what it means to be in a clinical trial. And we get a lot of questions about this. So I'm going to spend a little time about that and hopefully answer many of the questions that some of you may have in your mind about who are considering getting into any clinical trial. This isn't about our clinical trial. It's really about any clinical trial. Then talk about our program. First, a little bit about the NMDA receptor and GRIN-related disorders and an overview of our clinical trial. So who GRIN Therapeutics is, we're a New York City-based biotech company, and Nervati Neurosciences, of which I'm the chief medical officer, also oversees the company, and we have a single investor that's fully funded us through the approval, hopefully, of Verdiperdil. Verdiperdil is, you'll hear a lot about that. We're developing that for GRIN-related disorder with gain-of-function variants. Our clinical trial is underway. We'll talk a little bit about that, provide you an update. We were starting this last year when I presented. Company is a brand new company, formed in 2019, inspired by a GRIN conference at Emory University, and we've been the actual partnership with Nervati and the build out of the company began in 2021. And ever since, we've been working closely with the entire community, academic community, advocacy community. We want to make sure as a new partner in this area that we can learn as much from you and do things and address the needs that you're looking for. So now first, as I mentioned, talking about clinical trials. This is an overview. I'll go through each one of these individually, but it's really just a subset of the questions we get about clinical trial participation, but they're the most common questions. So I'll go through them individually. Sometimes it helps just to start with really what the most basic question is. What is the purpose of a clinical trial? A clinical trial truly is a research study. It's a research study that involves human participants. And generally, in the case of a drug development, it's evaluating experimental treatment. These experimental studies are carried out in humans after extensive laboratory and animal testing that shows that a potential treatment is promising, but also has an acceptable safety profile. The clinical trials are governed and structured by what's called a protocol, and these protocols go through extensive review by the health authorities. That could be the FDA in the US, different local authorities in Europe, as well as ethics committees that ensure that the study is appropriate for humans. I think one of the most common misperceptions about a clinical trial is that it is an experiment and therefore is not typical clinical care. It's often quite structured, compared to clinical care, which can be frustrating, and we'll talk a little bit about that. And this is a heavily regulated area where health authorities have regulations and guidelines that we, as a sponsor, have to follow and voluntarily do so, because it essentially protects participants from unreasonable risk. So a heavily regulated process that has a lot of structure, again, for the sole purpose of protecting participants. Now, we often get the question, if my child enters a clinical trial, will they see benefit? And really, maybe, but really, since a clinical trial is an experiment, there's really no guarantee that a participant will experience benefit. Part of the reason is that participants in many trials receive an inactive treatment. That's called a placebo, meaning there's really no chance of benefit from the drug. However, the close monitoring in clinical trials sometimes has an advantage. You have more interactions with healthcare providers along the way, so you're being watched much more closely than you might otherwise be in clinical practice. However, that also has its own burdens of being watched so closely. Your flexibility may be limited. And really, we always remind people that we're all super hopeful that a new treatment is going to benefit the participants, but often participation in a clinical trial benefits those who have the disease in the future. So the real contribution to this, which is a really meaningful contribution, is to those who come afterwards. So what do we ask in a clinical trial? They try to answer questions that primarily safety and efficacy. Is the drug safe enough to outweigh the risks related to the underlying condition itself? How should the drug be used? I mean, when you have a prescription written for you, it's often taken for granted. You take it this way, this often. Well, that's what you sort out in a clinical trial. What's the best dose? What's the best frequency of dosing? Are there special cautions that we need to learn about and then warn about in the educational materials that people get? And then, importantly, how effective is it? This is the big question. How effective is it at relieving symptoms? How effective is it at relieving the underlying condition or treating the underlying condition, slowing progression of the disease? And do those benefits outweigh any risks that are learned as part of the clinical trial before it's given to a lot of people, typically many, many more than are studied in a clinical trial? Another area we get a lot of questions about are these inclusion and exclusion criteria. And you'll hear these terms used quite frequently if you've been involved in a clinical trial. These are the strict rules of participation that have to be followed. Inclusion criteria are characteristics that a participant must have to get into the study. Exclusion criteria, characteristics that you cannot have to be able to participate. And to be in a study, the participant really must meet all inclusion criteria, and if they have any exclusion criteria, they cannot participate. And these can be frustrating, these could be challenging and lead to difficult conversations, but they're really there to ensure the trial is scientifically rigorous and minimizes risk, which is really this experiment. It's very important that it's done in a way that hopefully yields data that supports an approval, and not following these criteria often will lead to a failed trial, as we say, rather than a failed drug, because the study hasn't been conducted properly. So that's why they're there, and we know they're frustrating and disappointing, but it's the beginning often that these are more strict, and then as the drug moves along through development, they become a little bit more open and flexible. So now you sort of think, do I want to be in a clinical trial? What is the typical process? So the most important thing is to learn about the trial. There are, luckily, where we are with GRIN, is there are now options. There are people designing trials, promising approaches to this area of development. The most important thing is to learn about all the options and who they're for, and are they appropriate for me and my family. After you've gotten through that part, there's this screening visit, it's called, which is where the physician, the investigator, steps in. This is where the investigator looks at all of those inclusion and exclusion criteria to see if you as your patient or your child fits into this particular protocol. And there's a screening period where you have to sign informed consent, which I'll come back to, that confirmation of inclusion exclusion. And then the monitoring often of symptoms, sometimes seizures over a period of time to see if a participant qualifies. And sometimes, unfortunately, even though you've made it mentally to that point where you want to be in a trial and you go through all this process, unfortunately, the disappointment comes sometimes. You can't meet those criteria. And that means that for this trial, maybe it's the wrong trial, but that doesn't mean it's the way for another trial. So this is, we understand, a difficult thing if it were to happen, especially if you've gotten in that place where you want to be in a trial. But it's really, again, for the best of the participation to ensure safety. And there are going to be other trials and there are going to be other opportunities. And then once that's all confirmed, dosing and enrollment can begin. So I've mentioned informed consent a few times. This is probably the most important part of the process, where a participant or the family of the participant voluntarily confirms the willingness to participate. And that sounds very, legalistic, but it really is a documentation of understanding of all aspects of the trial, potential risks, potential benefits, and everything you need to know to make the decision for you and your family or your child. Often there's also a process called an assent, where a child is asked to at least acknowledge what's going to be happening to them if they're able. And then this is really done by written, signed, dated, informed consent to ensure that everyone is on the same page about this experiment that's about to happen. Now, thinking about a trial and what you should do or whether your family should participate, there's really no wrong answer to deciding here. Participation in a clinical trial is a very personal decision. It's not for everybody, and that's okay. But it's important to learn as much as possible, ask questions about the process, understand the time and the travel. As I mentioned, this is a structured process. Sometimes adds to the burdens of just general day-to-day life, and it's just an important decision, because the last thing you want is to sort of get in the trial and then find that it's too much and then have to leave the trial. And in any case, please make sure you're always keeping your child's pediatrician involved and keep him or her informed about what's going on, because it is part of the overall care. We want to make sure that participants in trials still stay in touch with their primary caregiver. We often get the question about travel between countries to participate in trials. This is really tough. Every trial is different. Cross-border participation in clinical trials can be very challenging. There, again, as I mentioned, a lot of rules and regulations regarding clinical trials. Every health authority is somewhat different. There's language barriers. There's the need to cross borders for visits. The duration, there may be a great trial that's only about 12 weeks long, but then there's a continuation phase, because it takes about 15 months to 2 years for a drug to get approved. You have to think about what happens if the drug works and I can't get the drug for 2 years. That's a big problem. There's also very strict rules about shipping experimental compounds across borders, which is too complicated to get into, but that's a big barrier. And then, of course, there's healthcare, insurance, general health. What if something goes wrong? There's a lot of issues with this. In any specific trial, it's something you can ask about and something you can discuss with your the investigator you're working with and the sponsor. And then finally, in the end, we as the sponsor and the regulatory authorities, we create all these rules, we create the structure, we work with physicians and experts on the clinical trial design and all of this, the protocol and all of the process. But every clinical trial site in every country has their own rules. So we defer to the investigators on who they believe is most appropriate. In fact, we can't even know who participants are because of patient confidentiality rules. So we, as the sponsor, have to defer to the sites. The study protocol has to be followed and reviewed and approved, but there may be different things at the site, including how they operationalize and how things go in terms of just the day-to-day conduct at the center that drive a lot of whether someone can be part of a trial. And as I said, we can't really be involved in any patient care, and to us, everyone remains anonymous. So often, if we're contacted with questions about a study, we will refer patients to different sites, and then we defer to the sites on how they handle those situations, because we are not allowed to be in medical practice. So that's my summary of sort of what again, common questions we get about a clinical trial. Certainly there are more, but we are always, when we're contacted about trials, we're always very open about sharing what we can and can't do. So remain a resource for the community. So in the last few minutes, I want to talk about our clinical trial. And that starts with understanding the NMDA receptor and why we are evaluating radiprotil in GRIN-related disorder. So the NMDA receptor is shown here. It's as a cartoon. It's encoded by the GRIN gene. And this is basically a receptor that controls the flow of calcium and has glutamate and glycine binding sites as shown here. And this is the target of radiprotil, which we will come back to. But to understand a little bit about radiprotil, we also have to understand a little bit about what gain of function is and loss of function. And often the receptor can be overactive. And we like to use this slide, many of you have seen this before, where gain of function means there's too much time open and loss of function is too much time closed. And we sort of use this example of a barnyard where you can imagine chickens in a barnyard, and you have a situation where a gate is stuck closed and the chickens can't get out and their normal passage out of the barnyard to get food. And so that is called loss of function. So that is a locked gate that inhibits the flow, while gain of function, you can imagine a broken latch where too many chickens get out or they escape or whatever. But that's the concept of the gain or loss of function, similar to when an NMDA receptor, which is like a gate on the membrane, is either open too much or closed too much. So when an NMDA receptor is overactive in GRIN-related disorder, this leads to the brain cells having trouble communicating normally. And this leads to the signs and the symptoms that you're all too familiar with epilepsy and neurodevelopmental symptoms, as shown here. There are no therapies specifically targeted for GRIN-related disorder. And as you know, often these symptoms are resistant to the treatments we currently have. And there really are no potential treatments for the neurodevelopmental symptoms. Now, radiprotil, now we're back to the NMDA receptor, and you can see, as shown on the left, this is in the setting of a gain of function situation. Radiprotil is designed to target a part of the NMDA receptor. It's what's called a negative allosteric modulator. What that means is it's meant to bind, as shown there, outside the receptor in the setting of gain of function to hopefully reduce this overactivity. And the hypothesis is that by binding to the NMDA, the NR2B subunit, which is this part of the NMDA receptor, in the setting of overactivity, you can potentially reduce seizures. And because this is also thought to be the underlying cause of the neurodevelopmental symptoms, hopefully modulate those symptoms also. So our trial is called the Honeycomb trial. It is designed to preliminarily evaluate these symptoms. First of all, evaluating safety and tolerability is always the first objective of a phase 1b study, and to identify the optimal dose range for future studies. However, we're also looking at the ability to measure impact on seizures, EEG, and a variety of behavioral symptoms and important symptoms such as sleep and motor function, to better understand potential benefit of Radiprazole. This is a study in patients with gain of function variants between ages 6 months and 12 years. And then we have two cohorts, a cohort of patients who have at least a seizure a week, who didn't improve after 2 other medications, or a behavioral cohort defined as people who have a significant behavioral symptoms. It's a global study. We have currently ongoing sites in Europe, and Canada and Australia, and recruitment is going very well, and we anticipate being able to share data later this year. So, a couple final words. Certainly, how to get involved. I mentioned up front, knowing about what's going on in this area, reading about our trial and others at clinicaltrials.gov is one way to do that, and using this as a resource. That clinicaltrials.gov list, as well as the UJAC CT in Europe, lists the requirements for these studies, so it's generally a pretty good place to start. Stay connected with your advocacy organizations, and do whatever you can to learn as much as you can, because knowing as much about the disease and what options are there for you is really the best way to make progress, support progress, and make change. As far as contacting us, we have a patient advocacy email address there, and we have a website that you can certainly feel free to reach out to. The other thing we're doing, I'll leave this QR code up there for a moment. Part of what we're trying to understand so that we can design our clinical trials better is to understand what is most important to the community in terms of what types of signs and symptoms, or what should we be designing studies to treat, and how can we better develop therapeutics for GRIN-related disorders. So this is a qualitative research study that you can, again, this QR code will take you to a place where you have to, again, meet inclusion and exclusion criteria. There'll be an informed consent, all the things I've spoken about, but it will bring you to a survey, and somebody will contact you about, again, interviewing. You may end up getting a survey, you may end up getting an interview about how we can better understand the experience of patients living with GRIN-related disorder. There's also an email address there. So in closing, first of all, once again, thank you to Cure GRIN for the opportunity to speak today. I also want to just point out that I'm just one person here who has the privilege of speaking, but there are so many scientists, collaborators, investigators that got our program to this point and continue to work very hard to advance and generate the data that's going to help us understand if Radiprazole works. And many of those are employees of GRIN Therapeutics and our CRO partners. We're a very small company. We have about 10 people, so we rely heavily on our partners and collaborators to advance this program. And most importantly, I have to thank the participants in our clinical trial. It takes a lot, as I hope I outlined at the beginning, a lot of work and commitment to be in a clinical trial, a lot of thought. And we appreciate that, and we don't take that lightly. So thank you again, and thank you to the entire community for adopting me and GRIN Therapeutics on this journey. So thank you very much.
Thank you so much, Dr. Panzar. We greatly appreciate your presentation, and thank you again to GRIN Therapeutics for sponsoring this event. I don't see any questions in the chat yet, but if you have questions for Dr. Panzar, feel free to add those, and we'll go ahead and transition to Dr. Garcia-Cazorla's presentation, and then we'll have time for questions at the end.
Hi, can you hear me?
Yes, we can hear you.
Okay, so thank you so much for the kind invitation to your meeting. I'm going to present the results of our clinical trial with Elserin in patients with green-related disorders, in particular loss of function mutations. So, well, just a few words about the importance of synaptic function in neurodevelopmental disorders. Well, there is a growing body of evidence that many disorders of neurodevelopment now have an origin in a synaptic dysfunction, and in particular, the glutamatergic synapses is very important because glutamatergic transmission is the most important type of transmission in the brain, and it's also related to processes of plasticity, excitability, which are really very important in neurodevelopment. So if we go to the structure of the NMDA receptor, there are different subunits, but which is important here for this presentation in the trial is that there is a specific site where serine, and in particular D-serine, will bind and activate the receptor. Glycine can also behave here in this site, and there are other sites of activations stimulated by glutamate, which is another type of amino acid. So the initiative of conducting this trial was based on a single case, on the first patient reported in the literature to have a benefit with serine in harboring a loss of function mutation. It was a green 2B with this mutation variant. In which during some experiments in the lab, our colleagues Xavi Altafaj and Mireia Oliveira could detect for sure a loss of function in this mutation. And then when giving deserine, and I will explain then why deserine in the cells and Elserine in the patients, they could observe a normalization of the current. So the conductance went again more or less by normal when introducing this amino acid in the cell and doing the neurophysiological studies. So that's why we tried to do the same in the patient, because we first started the experiments in vitro in the lab, but we use Elserine instead of D-serine because Elserine is a nutraceutical that is approved by the FDA and the EMA in Europe. It has been already used, and in particular in pediatrics, for different type of disorders and for inborn errors of metabolism, for genetic diseases that lead to defect in serine production. So this is why we decided, this is why it is the most appropriate molecule, the L and not the D, for patients. In addition to that, it has been reported that D-serine has nephrotoxic effects, or can have so some kind of adverse effects in kidney function. So that's why Elserine was the form utilized in patients, and also because Elserine is transformed into D-serine in the brain due to an enzyme that is called serine racemase, and then can behave in fact, as a neurotransmitter or activating the channel. So what we did in our first patient with this loss of function variant, in which the lab demonstrated this activation with Elserine of the transmission of the nerve impulse of the current, was to give the treatment. And what we detect, so that this child had a severe form of the disease at almost 6 years of age, with a severe hypotonia, inability to sit down, some stereotypes, also poor visual contact, and poor nonverbal communication, altered sleeping pattern. And then with 1 year and almost 2 years of treatment, what we detected is an interesting improvement with reactive smiling, with establishment of some visual contact and some gesture communication, was more interested in the world in general, and had quiet and autonomy to sit down, and was able to walk with some orthopedic walker. She also improved sleeping pattern, and epileptic form alterations without seizures remained the same. So we saw this improvement in this child, and that's why we wanted to start a clinical trial. So the ways is we've tried to see what was the best option to do that, and so because we just found this call, which is a competitive call to conduct research studies, in particular in inborn errors of metabolism, but also in other projects that have a metabolic approach. And this was the case of our clinical study, where we wanted to use an amino acid to treat this disease, that it is not a classic inborn error of metabolism, but because of this metabolic component in the treatment, we submitted the proposal, and we had a grant to conduct a clinical trial that was a very modest and like a small budget. And that's why we restricted our study to Spanish patients, because we could not open to beyond Spain because of these economic restrictions that we had. So this clinical trial called Tolerability and Efficacy of L-Serine in Patients with Green-Related Encephalopathies had the hypothesis that regardless the molecular etiology, and in patients with loss of function, green-related disorders could ameliorate glutamatergic function and improve the quality of life with L-Serine. And the primary outcome measures was, first of all, was to know about the efficacy of the treatment measured by change on neuropsychological and motor assessment, and also dose tolerability and safety. So we collected also the number of patients with any adverse event. This is the structure of the clinical trial. So we recruited patients older than 2 years of age, and we had a first visit, a screening visit with several tests that I will show you later on. And 3 months after, without any kind of treatment, we had the first visit and we started the treatment. And this period without treatment was just to capture a little bit the developmental history during these 3 months, because we don't have a complete and well-established clinical history, natural history for these disorders. So we could not compare with natural history, and we wanted to know which was the developmental outcome during these 3 months without treatment in our patients. So we started with 250 milligram per kilo per day, and 2 weeks after, we increase up to 500, and that was the stable dose. We had several visits, and then the patients stopped the clinical trial after 1 year of treatment. We used different scales just to assess adaptive behavior, like the Vineland test, also to assess verbal and non-verbal intellectual functioning, as you can see here, motor function, the gross motor function test. We also use sleep disturbances scale, behavioral, social, and emotional symptoms were also tested. Epilepsy diary and EEG were performed. Also, the quality of life was measured. So we developed and we set up a series of scales to measure all these functions. So regarding the results, 24 children were enrolled, 23 analyzed, slightly a little bit more boys than girls, and mean age was 10 years with a range from 4 to 18 years. And the most represented gene was GRIN2B, but we have also GRIN2A and GRIN1, and all variants were for sure assessed as loss of function variants. So our patients, most of them, almost 100%, had intellectual disability, and most of them from severe to profound. Behavioral disorders were also very common, 83%, and in particular inattention and impulsiveness, but also others. And then movement disorders, almost 80%, stereotypes being the most common. Epilepsy was found in 62% of our patients, and some of them, well, most of them have seizures, clinical seizures and EEG abnormalities, and some of them only EEG abnormalities. And here we can see that regarding refractory severe epilepsy, only 2 patients had this type of severe epilepsy. We had also 3 patients that had an EEG showing continuous spike and wave discharges in sleep. 2 were resolved before the treatment, and all of them were carriers of GRIN2A variants. So what about the primary outcome measures? So regarding the Vineland and different, aspects like communication, daily life score, socialization, and other aspects, what we could see is a difference between those patients who had a mild form. That means that Vineland score was greater than 55 points, and those with a severe form of the disease, a severe global hypotonia, developmental encephalopathy that was with a score below 55. So those with a milder form have an improvement in all the areas of the Vineland detectable, whereas those patients with the severe form did not manifest improvements. If we go to what we did is to not only to use the developmental quotient, but also the row scores of the Vineland, just to see if there were also changes according to that. And we also see even the statistical significant improvements in the mild group, in the group with the mildest clinical manifestations in expressive language, in personal autonomy, in all the abilities regarding how they get in touch and their relationship with the community, with their environment, interpersonal, so in different kind of abilities. Whereas in the severe group, these improvements were not found. So regarding the Bailey test, and the Bailey test is a test that is performed to test a normal development in children up to 2 year and a half, years. And here it is performed in general to evaluate children with developmental problems if the developmental age is below 42 months, more or less. But we can see that in the Bailey test, there was an improvement also for the severe group, for the cognitive domain at 12 months of treatment. And then we found also improvements in gross motor function and in quality of life for all kinds of patients, from the mild form and for the severe form of the disease. So it is something that is able to detect some improvements, not only in the mild form. Regarding epilepsy and how the treatment affect 2 children with refractory epilepsy, and one of them, the frequency of seizures decreased, and in 5 patients with intercritic abnormalities in the baseline EEG, but no clinical seizures, normalized the EEG during the trial. That's interesting. And we had also 1 patient who was seizure-free before starting the treatment and developed seizures during the trial. And this patient had a normal EEG baseline. We started with levetiracetam, and then the seizures normalized, and he was able to continue the clinical trial. We saw some improvements, nice improvements in writing and drawing. As you can see here, before L-serine and 12 months later, writing has clearly improved. And also drawing, in particular, the complexity of the drawing improve in many patients. And it is something that it is difficult to detect with the scales that we have, even with the Violin that detects many things, or other scales are not able to detect these kind of things. It is the same for these other aspects that also improve. We could detect that just in interviewing the patients, and also caregivers reported these improvements in imitative play and double meaning jokes. So they were able for the first time to start joking, and so that means that they had some kind of verbal irony. They could interact non-verbally better. The attention span and reaction time was better. Also, the executive functioning, the capacity of planning, organizing, initiating, self-monitoring, I mean, all these kind of things improved. And also there was a decrease in the number of nighttime awakenings, so the quality of sleep also improves. Regarding the adherence to the treatment was very good, as you can see here, and the reported side effects, you can see that 3 patients had some type of side effects. This patient had irritability, self-aggression, insomnia, hyperventilation. So we discontinued the treatment, and he was the one who didn't finalize the clinical trial. Then the patient that I already mentioned, who started with seizures, and after the introduction of levetiracetam, she was able to continue in the trial. And then a child with dyspepsia, that was something that due to a dose reduction could be solved without any other problems. And we also included patients that were in parallel following a similar similar protocol, although it was not exactly the same, but were in touch with us, and from different countries. They were patients between 3 and 14 years of age. 9 patients were recruited or were evaluated here, and eight green 2B and 1 green 1 loss of function variants. And 7 children had severe, profound, and 2 mild moderate intellectual disability, and with a severity of this intellectual disability directly correlated with the absence or highly poor oral communication. And despite the lack of standardized evaluation of these 9 external patients, and almost all of them had a severe phenotype, there were several positive observations, mainly in the verbal and nonverbal communication, as well as inattention, and adverse effects were very infrequent. So what we can see, what you know already, that the green gene family variants are cause of developmental encephalopathies with a spectrum of symptoms. That functional annotation is very important for personalized therapies, and gain of function now it has the possibility to start a trial with radiprudil, and loss of function has now this option of L-serine or other options, other positive allosteric modulator. It was well tolerated, and it was safe in our patients, and produced significant improvements in some domains of the adaptive, developmental, motor function, and quality of life. And subgroup analysis suggests that improvements in children from the mild group were greater, so these with mild symptoms. And it's interesting that L-serine normalized EEG patterns in 5 children, and the frequency of seizures in 1 clinically affected child, although this finding is to be confirmed in a larger sample. For sure, this work has just been published now in Brain, and well, all the team that contributed to this work is here. Well, the main responsible or people involved in the green team in Barcelona, Xavi Altafaj, Mireia Oliveira, who are the one doing the characterization of the functions, and also David Soto with the patch clamp studies, and then all the collaborators that we have in different centers in Spain. That you can see here, and that's all. And thank you so much for the opportunity of presenting the results. Thank you so much.
Thank you so much, Dr. Garcia Cazorla. We had two great presentations, and it looks like we have a bunch of questions in the chat. So we'll kick things off. Looks like our first question is for Dr. Panzara. It's related to the potential start date for participation in the GRIN Therapeutics trial for US patients.
Sure. So we are approved to be dosing in the US, and we're in the process of activating sites now. That takes time. Just stay tuned for that.
Thank you. Okay, we have a question about L-serine. So this person said that their child has GRIN1 gain of function and had a positive effect from that, but they didn't seem to have any benefits. The person was asking if it was because they're gain of function. Is that the reason why they didn't have much benefit, or is it something that they should try again?
Yeah, well, in general, if there is a gain of function, L-serine is not the most appropriate treatment because it will stimulate the receptor more than it is already working like too much. And so, yeah, I think that the patient has a gain of function. It is better to try some other treatments like radiprodil or some other blockers of the receptor. It will, yeah, will go better as an option.
Thank you. Someone else was asking about how L-serine is administered and how it's mixed or prepared.
Yeah, it's administered as a powder. We used Nutricia. It was sponsored by Nutricia, but it's also because in our country, in Spain, we can make a medical prescription of L-serine, just well, Nutricia, because it is a company that has been accredited for a long time ago as provider of medical food, and that in imbalance of metabolism is very reliable. So we can do that in Spain, and this is something that is covered by the public health system, and the patients don't have to pay anything. So, but well, regardless the company, the brand that is so involved in the production of L-serine. I mean, it's a powder that is given from 3 to 4 times a day. So the total is up to 500 milligrams per kilo per day. The dose that we use is based on the doses that have been used in other disorders that are imbalance of metabolism. However, this is something that is a matter of study, because maybe with lower doses it will still work. So this is something that we have to work on, just to have a better idea of what are the optimal doses. Yeah.
That makes sense. There was another question about loss of function versus likely loss of function versus possible loss of function. Did you guys distinguish between that? And if so, were there any differences in terms of if someone was eligible for possible treatment?
Was this to Dr. Garcia Cazorla?
Yes.
Yeah.
Yes.
Yeah, well, our experience in complex forms, maybe it is what the question means. I mean, sometimes it is not clear enough if it is a loss or a gain of function, because it is like somehow complex, and it gives not a like something very on one side or on the other. And when we have used L-serine in these forms, we have not obtained much benefits, or at least it has not been really something clear. And I don't know if these forms that are mixed or complex would work better with radiprodil. This I don't know.
Yeah.
It is also sometimes difficult to differentiate, so to know about the percentage of high and low in these complex forms. And that's what I can say. We don't have I mean, we have maybe seen, I don't know, 4 or 5 patients with these complex forms, but not too much, not too many. So we don't have a large experience.
I'll comment about the radiprodil. I mean, so for the gain of function studies with radiprodil, We have taken a very conservative view about what is qualified as gain of function. We base it on a publication by Myers et al. Last year that defines the criteria of what constitutes a likely or possible gain of function. And then we have an independent committee that looks at it to confirm possible or probable or likely gain of function. And the reason we do that is because it is a trial. And as we've said, we've taken a very narrow view for the purposes of the trial. Our hope is that as we learn more about these indeterminate or complex characterizations, that we then better understand about whether they could potentially benefit or where they belong in terms of gain or loss. But we're still not there yet, at least for Radiperdil, and we're continuing to ask that question. I think it's an important thing that everybody, as you're hearing, there are going to, as we learn more and more, there are going to be opportunities for gain and loss of function. It's trials. And I think it's great if people can start thinking about knowing what, how their variant is characterized, because opportunities may actually evolve. So that's one thing that people can think about is how do they find that out? Even if right now we don't have treatments available, but that could help them know if they're possibly candidates for a clinical trial. Something to think about.
Thank you both. It looks like the next question could be related to either of you. So did you notice any differences in outcome, I guess, thus far, related to the type of variant? So whether it be missense, frame shift, truncations, or position in the domain?
Mine's a quick answer. We don't have any data yet, so trial's still ongoing for Radiperdil, but those are the types of questions that I'm sure we will eventually look at.
Dr. Carzorla, did you guys notice any differences between the type of variant?
No, this is something that we would like to explore further, but we didn't notice a correlation between the type of variant and the response to L-serine. Yeah.
Okay. Let's see. Another question for you, Dr. Carzorla. The clinical trial for L-serine was for children age 2 and older. Do you think that there could be a benefit in starting children less than 2 years of age?
Yes, for sure. Yeah. I think that the earliest the better. So, and it is something safe. It is L-serine has been given to children also from the very first months of life in L-serine deficiencies. So in those inborn errors of metabolism that lead to a depletion in L-serine production. So there is no reason why we should not start also with at younger ages. Yeah, it was a question. We didn't have a younger patients, and also just regarding some safety issues at the beginning, we thought that it would be better to start with children older than two, but for sure in a patient few months of life with loss of function variation, I think that it will have a benefit starting with L-serine.
Okay, we have another question for you, Dr. Garcia Carzorla. Related to the L-serine trial, are there future directions to add a control group or to have a natural history study as a background distribution to rule out the improvement in the mild group, that saying that it's not just associated with age-related improvements? Also, were the presented significant results corrected for multiple testing?
Yes, we are planning that for sure. We are planning even with different options, different type of clinical trials that could have a placebo with, for sure. We are trying to do that. We are trying also to do some studies to optimize the dose. We would like to look at metabolic effects in general of L-serine, because it behaves when it is transformed into D-serine in the brain as a neurotransmitter, but then it has broad effects on metabolism, on energy metabolism, on the glycolysis, also in the lipid synthesis, sphingolipid production. So we would like to look at all these aspects, because in somehow they could also benefit, but we would like like to be sure that there are other aspects that are also like that everything is protected with L-serine, and we don't have an abnormal homeostasis in some metabolic pathways that are not that we won't like to find as results. So yeah, we are planning more studies. Yeah.
One thing I can add is that we're also planning to start a natural history study that will include all comers, gain and loss of function. The goal will be really to understand as much as we can about the natural course of disease. Also, I mean, I think what's been great about this community is the existence of a portal where variants are collected to be able to understand, to sort of help support that, and also to share with the community what we learned about natural history. And that's going to help to answer some of the questions that were just asked, both gain and loss of function. We just don't understand enough about the natural course of disease. And so we're now in a process of trying to get something like that off the ground. And hopefully that could serve as a community resource.
Exciting. Another question for you, Dr. Garcia Cazorla. Could you talk a little bit about the GRIN scale?
Oh, yeah. Okay. We are working on it, and it's almost finished. We're just in the statistical process, and yeah, at least we did that in Spanish because, well, it's our population first, and then it has to be translated and just shared. But yeah, it's almost finished, and I hope it could provide some help also for the clinical trials to measure some of the aspects that are difficult to quantify with the traditional scales, and be more adapted and personalized for GRIN-related disorders.
Thank you. A question for you, Dr. Panzara. Is Radiprodil a new drug developed specifically for the current clinical trials testing, or is it already used for other diagnoses or indications?
So Radiprodil is a pretty long history in that it was originally tried in peripheral neuropathy, diabetic peripheral neuropathy originally, but then was interesting that they learned of this specific targeting of the NR2B subunit and its function as a negative allosteric modulator. And that led to some folks, our founder, Andrea Muglia, and others who said, let's try this in GRIN-related disorder. And actually, it was started initially in infantile spasm by Stéphane Aulin. And then basically some of those encouraging data are what prompted Nervati to work essentially with Blackstone Life Sciences, our sponsor, to acquire GRIN Therapeutics and start this program. So that's sort of the history, is some promising, excellent safety data, another indication, a novel mechanism of action discovered along the way, a pivot to a new disease area, kind of a nice story about how sometimes there are those drugs out there that people don't think are past their lives, but you find a new life and something exciting to explore.
Thank you so much. Okay, I think we'll end on this last question. Dr. Garcia Cazorla, did you notice any changes in muscle tone with L-serine in any of the patients?
Yes, it was detected in the based on the results also of the gross motor function scale. But when we visited the patients in every control visit and the clinical examination already detected some improvement in muscle tone, in global hypotonia, but also in muscle strength. And yeah, sure, yeah.
Perfect. Okay, one more question I see here. So if a child started taking L-serine and had positive results, would they need to keep taking it indefinitely to continue to see the said results?
Yeah, this is something that we also would like to work on, because what we have seen when patients stop L-serine is that these improvements remain stable over a certain time. So does it mean that we should give L-serine for certain periods of time and then have a stop for a while and then retake again, the L-serine, this is something that we still don't know, and we are working on it. Yeah, but it is a very, very interesting question, and we also are, well, yeah, we would like to know the answer.
Wonderful. Thank you both so much for your time, and thank you everyone for your attention, your engaging questions. We greatly appreciate it, and all of the exciting results that are coming up, as well as future directions and exciting plans. With that being said, we'll go ahead and close up the question and answer period, close up the session, and I believe we are currently in time for a break, and then we'll transition over to our keynote. So thank you all so much, and we'll see you soon.
Thank you very much.
Thank you very much.
Good
Bye.
question too. Thank you. Bye-bye.
Bye-bye.